Hello and welcome to this virtual organic chemistry lab. I'm Derek France. In today's video, we're going to do what I think is the coolest experiment that we've done this far in the lab. We're going to take this compound right here, 2-naphthol, you can see its structure, 2-naphthol, and we're going to deprotonate that alcohol group with sodium hydroxide to generate the naphthoxide, the O- on the naphthalene structure, and we're going to take that alkoxide, treat it with bromobutane, and perform an SN2 reaction. So this will be our first synthetic organic chemical reaction that we're doing in the laboratory. I'm in a great mood. I love this lab, so let's get started.
Before we carry out this experiment, let's talk a little bit about what's happening theoretically in the reaction. So we are going to synthesize 2-butoxynaphthalene, and this is the literature reference for the reaction we're going to run. It's from McNulty and coworkers. It was published in the Journal of Chemical Education in 2009. So if you want to go check out that reference, there it is. So this process is going to be two steps. In step one, we're going to have a proton transfer, which is really just an acid-base reaction. We're going to take 2-naphthol, and I'm indicating this proton right here in green because we're going to be removing that proton in an acid-base reaction. To the 2-naphthol, we will add sodium hydroxide. So we're adding sodium hydroxide, which is a strong base. This will generate an equilibrium reaction in which the hydroxide right here will deprotonate the proton that's connected to the naphthol. Let's draw the result of that process. This is the proton transfer step that will occur in step one of this reaction process. Naphthalene will be deprotonated by sodium hydroxide to generate this sodium naphthoxide right here with a negative charge on the oxygen atom. And we'll also generate water, so the hydroxide will become water after the proton transfer occurs. So let's think about which way the equilibrium, in which side of this equilibrium we're going to favor. Is it going to favor the products or is it going to favor the reactants? So if we consider what is the pKa of this proton right here, the pKa of that proton is about 9.5. So it's much more acidic than a typical alcohol. Normally alcohols appear, they have pKa's about 16. This is much lower, about 1,000,000 times more acidic, and that is because this ion right here can be delocalized through resonance. I encourage you to draw all of the resonance structures for the naphthoxide ion. Take a moment to do that if you wish. But because of the resonance stabilization, the delocalization of that negative charge in this ion, the naphthol has a lower pKa, a higher acidity than we would normally expect for an alcohol. On the other hand, water over here has a pKa of about 15.7. Water is not as strong of an acid as the naphthol. So because of that, you can do a KEQ calculation. I'm not going to do it here, but you can certainly do that. Take a moment to do that if you want. But what we can tell, because the pKa is lower on the left side, the strongest acid on the left side has a lower pKa than the strongest acid on the right side, we're going to favor the products side of this equilibrium. So this side over here will be favored. We're going to generate a lot more of the naphthoxide. And if we did the KEQ calculation here, I encourage you to do that, we will get enormously more of the materials on the right side than on the left side. Now that this naphthoxide ion has been generated, that's going to be the nucleophile in step 2, which is our SN2 reaction. In the SN2 reaction, we're going to begin with this naphthoxide as our nucleophile. And to this nucleophile, we're going to add 1-bromobutane. 1-bromobutane is a great substrate for SN2 reaction. It's a primary halide, and our SN2 reaction can occur through the typical SN2 mechanism. where the alkoxide attacks this carbon right here, and then bromine. The bromine leaves as a bromide ion as a leaving group. So that is our SN2 reaction. The product here will be 2-butoxy-naphthalene. That will be the product of this reaction. And let's think about what other products we might get. We're going to end up with Br-, and that Br- is just going to be associated with the sodium ion that has been hanging around as a spectator ion this whole time. So we will generate sodium bromide as a salt during the course of this reaction as well. To accomplish this reaction, we are going to use a solvent. That solvent is going to be ethanol. And we're going to be heating the solvent to, I should say the reaction mixture, which is mostly solvent with this material dissolved in it. We're going to be heating that to reflux. And that reflux just means boiling. We're going to heat it to boiling. The ethanol will also be present in the first equilibrium up here. So we're going to have ethanol as our solvent for the proton transfer step, and we're going to have ethanol as the solvent for the SN2 reaction as well. So this is what we're going to do theoretically. Now let's look at practically how we're going to carry out this experiment in the lab.
Okay, here is our experimental setup. We have a 100 milliliter round bottom flask right here. It's connected to a vertical post. We want to make sure it's on a vertical post, that way it doesn't start dipping during the course of the reaction. Look how well the clamp is secured. A 3-finger clamp with the top finger right here, right under the lip of the flask. That is the most secure way to hold your flask. Down at the bottom of the flask, we have a couple boiling chips. That's just going to allow the solvent to bubble. Normally, we would put a stir bar in there, a magnetic stir bar, and then below the thermowell, we would have a stirring plate, a magnetic stirring plate. But sometimes we don't get enough magnetism to flow through the thermowell, and it doesn't stir effectively. So we're using the boiling chips because as this material boils, that will create enough mixing to allow this reaction to take place. Thermowell will be applying heat to the reaction, and then we have this attached inline variac that's going to control the amount of current that goes to the thermowell. If we directly plug the thermowell into a wall outlet, that's going to get extremely hot extremely quickly. So the variac controls the current, controls the amount of heat, and allows us to monitor the amount of heat that we're providing and control it. Above here, there is a condenser. So we're going to be refluxing or boiling the mixture. The vapor phase will be condensing in the condenser and then falling back down. The condenser prevents the reaction from just drying out. We don't want that. We want to keep the solvent in the reaction. So it's going to bubble out, condense, fall right down. The blue line, the blue tape is indicating that this line is cold water. And then the red tape is the exit. So the water's flowing in from the bottom, exiting out the top. Why do we do it that way? There's a reason why we do it that way. It's because if you fill from the bottom and go out the top, you fill this entire jacket with water and maximize the surface area for the internal tube to be cooled by that cold water. If you just flow from the top, you're just going to have this waterfall, and the water is going to come out of the bottom, and then the jacket's only going to be partially filled. Filling from the bottom allows for full filling of cold water in that condenser. But we don't want to have that on there yet. We need to add our ingredients, our reagents to this mixture. I like to use the word ingredients because I like to think, ingredients isn't really the word you should use, but I like to think of running an organic chemical reaction as cooking, except you just don't eat what you get. You don't eat the product. You view it and you admire the product, but you don't eat it. Here we have our reagents. That's 2-naphthol, 1.00 grams of it. That's sodium hydroxide. Zoom in here. That's sodium hydroxide, 0.56 grams. look, it has kind of a glistening appearance. That's because it's very hygroscopic and it's picking up atmospheric water to create that glistening surface. But don't let that beautiful appearance fool you. That is nasty stuff. If that gets on your skin, that's going to burn like crazy. So be very careful with the sodium hydroxide. And then right over here we have our solvent, which is ethanol, 20 milliliters. So for the first part of the reaction, we're just going to do the deprotonation of the naphthol with sodium hydroxide. This is setting up for the SN2 reaction that we will do in the second step. So let's start setting this up. Okay, let's set up the first part of the reaction. Again, the boiling chips are already in there. To this flask, I'm going to first add the sodium hydroxide pellets. I'm not going to use my funnel for this because the funnel I have is a little too small. And look, because it's so hygroscopic, it actually sticks to our whey boat. So it's actually, look at that, it actually kind of stuck to the glass right there. You see that? So I let it, in the context, or not in context, in the process of making this video, I had to let it sit out a little bit longer than I would have liked. But there we go, it all fell down to the bottom of the flask now. If you look, there's still some liquid here. Be very careful with that liquid. That's super concentrated sodium hydroxide. So we want to set that off to the side and make sure our finger doesn't touch that. There's a little bit up here that we will rinse down when we add our ethanol solvent. Now we can take this funnel, add it to the top, and we're going to add the nap-2-all, the 2-naphthol. This had a couple large particles that I broke down with a spatula, and hopefully it will all flow down through here. If it gets stuck, I have to break it up with the spatula. See, it's kind of getting stuck. So just break it down so it can go through. All right, try to get as much off of that whey boat as you can. Okay, there you have it. So if you take a look at our funnel here, there's still a lot of our compound, our reactant that we want to make sure gets into that flask. So we're going to rinse as we add our solvent. It's always good to add your solvent last. We can rinse down the sides of this funnel to rinse all of the materials down into our round bottom flask. Okay, and I'm just going to use this funnel. I'm going to lift it up a little bit here. My hand is in your way. Lift it up a little bit here so it also rinses off some of the sodium hydroxide that got stuck on the side of the top part of the flask. Okay, there we have it. Now all of the materials for the first part of our reaction have been added. As I said before, usually you would have a stir bar in there and that would just start stirring, but we don't have that for this particular experiment, so I'm going to take it out, give it a little swirl, try to get as much of these materials to dissolve as will dissolve. Look, we have the development of a greenish color. It kind of looked brown when I first added everything, and now it's turned green. And if we look carefully here, I don't know if you can see it, we can see that, there you go, you can see that there are sodium hydroxide pellets still in there. The naphthol has pretty much completely dissolved as far as I can see, and the sodium hydroxide pellets are still there. So for the first part of the reaction, even though proton transfers generally happen pretty quickly, we're going to heat this up and let it stir for, not stir, let it reflux for about 15 minutes, and we're going to just do that so that all that sodium hydroxide gets dissolved and can react. Before we begin with the heat, we want to make sure we place our condenser on top. And now we are ready to heat. I haven't plugged in my variac yet. Now we will. So I'm going to start cranking up the heat here on the thermowell. And we want that thermowell to just barely be touching the flask. So we're going to let the reaction sit here and heat for 15 minutes. Okay, it's taken about 5 minutes to get to this point. You can see the reaction mixture is starting to boil. And the boiling chips are the site of the bubble formation. So if this starts getting really out of control, you want to lower the lab jack to quickly reduce heat. But we have a nice reflux going now. We can see that the droplets of ethanol are collecting here in the condenser and then dripping back down. So you can see those drops. That's ethanol that's collecting up on the condenser and dropping back down into the mix. Already looks like a color change has occurred. It's getting more of a brownish tea colored appearance. I'm going to keep an eye on this, make sure it doesn't start going too crazy with the boiling. But that's a good reflux right there. And we'll let that continue for about another 14 minutes. Our reaction has now been running for 15 minutes. I'm going to lower the lab jack and allow the bubbling to stop. We don't want that to be bubbling. We're going to be, in the next step, removing the condenser from the top and then adding our bromobutane, which will be the substrate for the SN2 reaction. Okay, at this point, the boiling has subsided, so we may now remove the condenser from the top of the flask. Just kind of set that off to the side. And right here we have 1.0 milliliters of 1-bromobutane. So the 1-bromobutane is our substrate in the SN2 reaction, and the naphthoxide that we have just generated is going to be our nucleophile. So now we are ready to heat this back up by lifting the jack. And our SN2 reaction will happen now. And we're going to let this reflux, or I should say, let this reaction stand at reflux for about an hour. You can see the thermowell was hot still, so as soon as it touches the flask, we have reflux occurring yet again. And we will come back and check on this reaction in an hour. Of course, we are keeping an eye on it to make sure it stays safe. After only about 5 minutes, we see what appears to be a color change in the reaction. That used to be a lot darker brown. Now it's a little bit lighter brown. Not much that we can, say here, but at least we can see that something is happening. There's an appearance of some type of change in the reaction. So it's been only 5 minutes. We're going to let this reaction run for an hour, so another 55 minutes, and we will come back. Our reaction has been running for about 40 minutes at this point. We still have another 20 minutes to go. And here you can see that it's definitely gotten lighter in color. So again, about 20 minutes to go in this reaction. Okay, we have reached the hour mark. At this point, I'm going to turn off our variac so that we're no longer going to be supplying heat to the thermowell. We can go ahead and remove the thermowell by lowering the jack, and we can take off the condenser. Once that stops boiling, I'll take off the condenser, and then we're going to let this reaction just sit here. It's going to sit for as long as it takes for the reaction to become the reaction mixture to become about room temperature, easy to handle, not hot to the touch. Our reaction has now cooled down to room temperature. I can touch here, it doesn't feel hot. So at this point, we're going to take just a couple drops, just like that much. Take a few drops out of this mix and put it in a vial. And we're going to just save this for a TLC experiment that we're going to do later. At this point now, we are ready to collect our product from this reaction. Over here, I have a 250 milliliter Erlenmeyer flask that has 25 grams of ice. So that's just ice. And we're going to add this mixture, this ethanol solution, which has our product, as well as some other things. And we're going to carefully add this to our Erlenmeyer flask. So we'll add a little bit at first. Swirl, make sure we swirl this around. Kind of looks like a creamy drink of some type right now. We'll add more. Keep stirring, keep swirling around. And then finally, I will add the rest. I'll set that reaction flask up here for now. And we'll keep swirling our mix in ice. And we want to make sure we get as much of this mixture out of here. So I have some cold water that has been on ice for a little while. We can add that. Should be using a funnel here, but you know. Add that, swirl, make sure we get all of the contents of the round bottom flask into our Erlenmeyer flask. Okay, and so this is our mixture. You can see that on the side of the flask, some solid formation. There we go, now we're focused. You see the formation of solid. That's not ice. That's not little ice particles. That is going to be the product of this reaction. We are precipitating it out. So the product of the reaction is soluble in ethanol, but it's not soluble in water. But it does have a melting point of about 30 degrees. So we have to get it nice and cold into this water mixture in order to form a solid suspension of the product. Now we are ready to perform our vacuum distillation. We have our Buchner funnel here with our filter flask as well as our vacuum adapter and a filter paper inside the Buchner funnel. I'm going to turn on vacuum. And now that has sucked the paper down so it's nice and flat, but we should always just add a little bit of our solvent, in this case water. This is some nice cold water that's been on ice. That will press the paper down even more. And here is our mixture of water as well as the ethanol from the reaction and the solid, which we are presuming to be the product that the organic product of the reaction. So we're going to carefully filter this material. And we want to get as much of that solid off the side of the glass as possible. So with cold water, we will rinse. So we'll rinse several times. You can see it's kind of stuck. We might not be able to get it, but we'll do our best. Okay, keep going here. All right, so with a few rinses, we were able to collect our product. I'll turn the vacuum off for a moment so that we can pull this off, and you can see there is our product on top of the filter paper. A nice sort of off-white solid. That black thing over there, that's just a piece of our, that's one of our boiling stones that remains in the mix. And now we will analyze this product to see if it is pure 2-butoxy-naphthalene. Now we are going to analyze how well this reaction worked by using thin layer chromatography. Our solvent system is going to be 25% ethyl acetate and 75% heptane. On the left over here, I have a sample of our NAPTH2-ol, the reactant for the reaction. In the center, I have, this is a few drops of reaction that we took out before we did the vacuum filtration. And over here, this is the product that I obtained from the top of the Buchner funnel. So by comparing these, we will be able to see how far the reaction went at this point, how much of the starting material, the reactant, is left, and then based on how this spot looks, we will be able to tell if our product is a pure product or if it still retains some of the original reactant. So let's spot each of these, and we want to be sure to use different capillaries for each of these spots. Okay, so the spots are on the TLC plate. And so under UV light, we can see that we have enough material on there to see good dark spots. And this should give us a good TLC. So we will put that in the TLC chamber and see what we get. Okay, our TLC plate has developed in the chamber, and now it is out. We are ready to visualize with the UV lamp. Take a look at that. Let's remember that the left side here, over there, that spot is naphthol, NAPTH2-ol. That was our reactant. The center here, this sample that now has 2 spots, that was the reaction after 1 hour of reacting. When we cooled down the mixture. And then finally, this spot right here on the right side, that is the material that we collected from the vacuum filtration. So I want you to answer 2 questions. 1, was the reaction completed after an hour? Second question is our product pure? The product that we obtained by vacuum filtration, is it pure? Did you think about it? If you're not done thinking, pause the video and think about it. Okay, I'm assuming you paused the video. Here we go. It turns out that after an hour of reacting, our reaction was not completed. We still have this spot, which is the same as our starting material. So there was still a substantial amount of 2-naphthol in the mixture. So we were not at completion, although we did have a substantial amount of product as well. During that precipitation that we did, we were able to precipitate out our pure product, and we do not see any naphthol in that product. So the naphthol remained soluble in water because we had base present, but the product was able to precipitate out. Okay, well, this shows that our reaction gave us a nice pure product after the vacuum filtration, but we still have a substantial amount of unreacted starting material in the reaction, and that's going to definitely lower our percent yield of product. Using the power of organic chemical synthesis, we were able to transform 2-naphthol right here into this product right here. And at this point, we don't know the identity of this product. TLC tells us that it's certainly not 2-naphthol. It has converted into something else, and based on the TLC, the fact that it had a higher RF value means it's not as polar as 2-naphthol. But in order to be 100% sure that this is 2-butoxynaphthalene, which is what we expect it to be, we will have to run an IR spectrum, we will have to get its melting point, and most importantly, we will have to run NMR spectra. That will not only tell us if it is in fact 2-butoxynaphthalene, it will tell us if it's pure as well. So that's all we can do at this point. We can't be 100% sure that this is 2-butoxynaphthalene until we get those spectroscopic characterization data. Now, at this point right now, we would have to let this dry out. It's still wet from the vacuum filtration. We would get a yield to see how well the reaction proceeded, and then get that spectroscopy. But that will fall outside of the scope of this video, which focused purely on the synthetic procedures to obtain the product. Okay, well that wraps up this video on the synthesis of 2-butoxynaphthalene via an SN2 reaction. And I'm Derek France, and I look forward to seeing you again in Virtual Organic Chemistry Lab.